Well, hi, everyone. Thanks for having me. Thank you, Amanda, and Dr. Mason also for inviting me to come give a talk. It's a real kind of pleasure to be here, and a museum person myself. I certainly have enjoyed coming to some of the talks and getting to know all y'all a little bit. Anyway, yeah, I'm going to tell you a little bit about myself and my research today, and I'll kind of start with a slide progression. There we go. I always kind of like to start with this image right here in some of my talks because I feel like to some extent this captures a lot of my motivations in terms of my research. So this is in South America, specifically in a small country called Suriname up in northern South America. And within Suriname, specifically here within the Sipilowini Savannah on the border with Brazil. Now, what am I actually doing here? If I draw your attention to these rocks right here that I'm sitting on, you'll notice a very thin film of water. And I am here looking at this. This is what's called a hygropetric seep. This is basically any place in which you have a little bit of water coming out of the earth or some kind of substrate and forming a very fine film with an algal mat and that kind of thing. And I'm here looking for a beetle. This particular beetle that I'm looking for is Notomicros. This is a species that I described back in 2018. Petroreptans meaning rock crawler appropriately because guess where you find it. Now, how does it actually exist in this tiny film of water? Well, Notomicros, as you might have guessed from the name, is very, very small. Next time you find a penny on the ground, pick it up, look at it, and you might think, Dr. Baca's beetle is the size of that little L there. Isn't that tiny? What is he doing? So, kind of sitting here looking for this thing, naturally you need to give your eyes a rest and taking a little break, kind of taking in the view. And at least for the purposes of this talk, you kind of look out. I like to think that I was looking out the landscape and thinking about Notomicros petroreptans and the other diversity that's kind of around, and not only what else might be out there, but also thinking about how did it get here in the first place? You're kind of on this little isolated island rock poking up out of the earth, a little bit of water trickling down, and you find beetles in it. Well, what is it actually doing here? And that really is sort of the heart of a lot of the research that I do, is that I'm trying to understand what is the full extent of aquatic beetle biodiversity, and also trying to understand the drivers of that biodiversity. And specifically, I really like to focus on how geography and landscapes influence diversification in this group. And South America, of course, gives you quite the diverse landscape to do this in. So this is a 3D render of an old USGS map that's done by Sean Conway. Really, really cool map. And I think it really, really sort of just drives the point home of just how diverse South America really is. It's not just the Andes and rainforest. There's a whole lot more to it. And just to kind of orient you here, I am sitting in this little area right here, again in Suriname, the Sipilowini Savannah, which is kind of in the heart of what we call the Guiana Shield, which is this uplifted region up in northern South America. I'll be talking to you a lot about that today, kind of as we go in, so just keep that spot in mind there. And for the outline of my talk, I'll give you just kind of a quick introduction of the aquatic beetles that I actually study. And then I'll have a couple sort of quick research stories focusing on the aquatic beetles that you find in South America and the Guiana Shield. And then finally, just talking a little bit about some of the big picture patterns that I'm sort of seeing and how that ties into my work here at LSU. So first, aquatic beetles. To sort of give you a little bit of a foundation to start with, the thing to kind of know about beetle diversity is that it is vast. So first off, insects make up half of all the known organisms on the planet, right? There's about close to 1 million described species of insects. And beetles make up about half of that, meaning that beetles are about a quarter of every described thing on the planet at about 400 to 500,000 species. And that's just the known ones. We estimate that that's about a quarter to only half the actual species that are out there. So our work is quite cut out for us. Now, my favorite beetles are the aquatic beetles. And they come in a whole kind of wide variety of shapes and sizes and a lot of different specializations. And just to kind of give you what they look like a little bit as well, when you actually see them in the wild, they don't really look like much, but they actually kind of, have a whole wide diversity in both form, function, and just actual biodiversity. in all themselves. So you want to kind of see what we, this is kind of us how we catch our beetles when we're out. Just kind of give you a little example of what you might find in the fine. And these are all aquatic beetles and string. But yeah, so this is in the Llanos of Venezuela, which ties in a little bit to what I'm going to be talking about too. Now, one thing to know about beetle evolution and aquatic beetles in general is that this really isn't describing any sort of natural group or a clade. It is actually more what you would call an ecological guild. There have been about 8 independent invasions of beetles into the aquatic habitat. And so far, we have about 30 or so families spanning 3 suborders. And aquatic is also a term that I kind of have to use a little bit loosely. It's like, how do you actually define aquatics, right? It's like, well, you might think of some general habitats like your ponds, marshes, maybe some streams or waterfalls, and even kind of the strange hygropetric seeps that I talked about. And naturally, the aquatic beetles that you find there are maybe what you might expect from one. These kind of streamlined beetles with these really nice swimming adaptations and whatnot. But it gets a little weird. For one, is a lot of aquatic beetles have actually transitioned back into the terrestrial habitat. One particular example is this genus here, Quadriops, that lives on rotting fruit. For a long time, we had no idea that that was the case. It was described from a couple of samples from a flight intercept trap, had decades of really not knowing what it was. And then randomly, when I was in Guyana with my advisor, he picks up a rotten Clusia fruit and is like, oh, look, there it is. And like, cool. And then you also can find some other weirdos that live in places like underground aquifers. So you find these all over the world, especially in xeric environments. So, you think like Texas, Australia and whatnot, is that you'll actually put a well tapping into an aquifer underground and beetles come out of it. And that has evolved independently many, many times across different beetle subgroups. Now, I specifically study the family Noteridae. Now, these are what you would call the burrowing water beetles. Commonly, you probably have never heard them by either name, but either way, there you can take your pick. They are not a big group in terms of beetles, nor a big group in terms of size. The biggest one is still less than a centimeter long, and the overall diversity is only about 280 described species, which is rather small by beetle standards, certainly. And even again, going back to this pan and kind of look, is that you might not even know you're looking if you happen to see them. So this is species of this genus Panthicellus that I described, and they just kind of look like little black dots. What's fun is that once you actually do this enough, you can tell what they are. Just it's like, oh yeah, that's Panthicellus. There goes. But once you actually get them under scope, there is a amount of diversity to be held, and it's kind of again getting after the drivers of this diversity that I'm trying to. And I've done this through a very integrated research approach. So I do a lot of documenting of biodiversity, and that's just through general taxonomy. It's where I got my start at the University of New Mexico in terms of just doing generic revisions and describing new species. I then use that information on the biodiversity to actually, help studies that are looking at evolutionary patterns. And this is, phylogenetics and genetics. And then finally, I use both of those together to understand evolutionary processes and try to tie in hypothesis testing to actually understand what might be the causal drivers of both the biodiversity and patterns. And naturally, these are both very integrated. And I've also done these across many different time scales. So going all the way back to the Permian about 250 million years ago and looking at very, very deep relationships in the parent clades of my beetles, and then also going all the way up now into the species of population level, which is where most of my research is actually taking place. And so that's what I'm going to really be talking about today when I kind of get now here into this, neotropical phylogeography in terms of trying to understand some of these drivers within some of these specific genera. Excuse me. So I'm going to go back to Notomicros, the one that I introduced to begin with, this really, really tiny thing. Kind of what you need to know going into this, they're all very minute. And there's only about 18 described species, though there are many more undescribed. And here's kind of a look at just a few of those. You'll note that about 2/3 of these have names on them, while the rest of them no longer do. And these are just undescribed species that I happen to find in from our field work and also within collections. They have a very disjunct distribution and are familiar with biogeography might actually recognize such a distribution. You'll find them in both sort of the Western Pacific, in the Malaya, Oceania, and then you'll find them throughout the New World, and especially in the neotropics. But there's this really big biodiversity gap, and this is kind of getting into the heart of the questions I'm looking at and trying to understand is why is there this disparity in biodiversity? If you look at the Old World, there's only 2 described species there. And I can say that to some extent, when you look at these, at least from like a morphological standpoint, it really only looks like there's 2 species there. Whereas when you go into the New World, you see many, many more species, and there's just a whole lot of more morphological diversity also there. And so I'm trying to kind of understand exactly why there's this disparity in biodiversity. It went ahead and just did a quick Sanger, right? I put together a Sanger phylogeny, and just to show that these lineages were actually reciprocally monophyletic, right? So basically that means that there's no recent migration, that any of this diversity isn't being driven by any sort of like cross-ocean migration or anything like that, that these are each other's, that these are both independently evolving. And then we went ahead and actually threw a divergence time analysis at this also to show the timing of the actual divergence of these groups and found that aligned very, very well with Gondwana and Vicarians. And so then as the tectonic plates, drifted apart, you have South America and Australia separating from Antarctica. And then you end up with these sort of, divided clades evolving on their own on opposite ends of the world. And so with that, looking at New World Notomicris, we can kind of, get at this question. Okay, we're fine. We can kind of get at this question now, I think, with a little more confidence that we're actually looking at just one clade. And I decided to go ahead and kind of, when thinking about this sort of, diversification and how it ties in with geography, I really go back with me sitting on this rock, thinking about the really complex geography of South America and wondering how it's driving this group. And so I really kind of wanted to approach with this hypothesis that it is this geography somehow playing a role in driving the diversity of Notomicris. I focused on one particular clade for this, the Notomicris trail-eye group. And I picked this group for a couple of reasons. First, there are a few described species in it, but the morphological delimitation is actually really, really subtle. There's no real clear patterns in morphology that help you separate these species apart. And so it's really tough to actually tell the difference between interspecific and interspecific variation. So part of my, reason for picking this was just to trying to understand how to actually delimit this diversity from a tactical point. And then they're also very widespread in the neotropics and, of course, monophyletic, which means that they're a good system to go ahead and actually test for some of these geographic drivers at a shallower scale. And furthermore, is that they have what appeared to be a lot of overlapping distributions in the Guiana Shield in separate lineages. So you can see those highlighted up there on that picture. And so these are a couple of the questions that I'm kind of going after, and just kind of to sort of orient you a little bit. The estimated crown age of this whole group came out to about 13 million years, which I know by some standards is a little bit old, but we'll be looking at a little bit more shallow scale as we go forward. To do this, I went ahead and took a phylogenomics approach. So in grad school, I went ahead and built a tailored UCE probe set targeting specifically notarids using model genomes from across the notarid phylogeny. And targeting about 3,400 UCE loci in total. I picked those across the whole trail eye range with a big emphasis, though, on the Guiana Shield and included museum specimens where needed. And we did pretty good in terms of the actual locus recovery. And we got a very robust phylogeny from that. So we were really, really happy with these results. And what we did find is that a lot of these overlapping distributions in the Guiana Shield were actually real. So this wasn't just some, from a couple of markers, kind of given us weird topologies or anything like that, from inferred from about 1,000 loci, we do still see this pattern of distinct lineages showing overlapping or sympatric distributions. But this question is still like, okay, is this like population structure, or is this actual showing genetic distinction when you're looking at this phylogeny? And so that's kind of why we stepped a little bit here into population genomics, went ahead and harvested some SNPs, and went ahead and threw some clustering methods at this problem also. And what I can say is, as a take home, is that the clustering and population assignment methods that we use for this were really consistent with the phylogeny and showed a lot of genetic distinctness and with very, very little admixture. And let's see, I think I have a slide. There you go. And again, within these particular groups that you actually find with these sympatric distributions or very close distributions in the Guiana Shield. And to take a look at kind of like a particular one that where you even have, in this case, sister groups, which are genetically distinct, but, you know, completely overlapping distributions here. So kind of the take homes from that is that there are many genetically distinct lineages, but likely several undescribed species that we still have yet to find from this. Now, I'm kind of thinking about, though, the patterns and processes is that when you start looking at this and kind of thinking about, okay, is the Guiana Shield actually promoting diversification? You get this pattern that you call phylogenetic overdispersal. So this just pretty much means that like the lineages that you find in one place are from all across the phylogeny. So it's just describing this pattern that you're seeing right here, right? Now, this is consistent with a process that is dispersal mediated diversification. Because basically you have these beetles that are kind of going back and forth from these uplifted regions. You have reproductive barriers established. And then even if they make it back into the Guiana Shield, what ends up happening is that they're reproductively isolated. You have isolated lineages, right? But the problem is that also could just be that it's really, really conducive to lineage survival. It's possible that these are just really widespread populations, but then because of the volatility that South America is known to experience through, say, climatic fluctuations and other changes, you know, South America is a very, very dynamic place. What is known is that the Guiana Shield is just less volatile. So it's possible that rather than being something that's dispersal mediated on its own, it's just that lineages are just way more likely to survive up in the Guiana Shield. And trying to tell the difference between those two things, or I should say the relative contribution of those things, is really, really difficult to get at. But it is something I'm kind of interested in, and so I don't really want to stop there. Like the real take home of that is that this isn't, you know, an end point. This is just a starting point. And the first thing that I want to do is start balancing out some of our sampling in this group to get a better feel of some of these dynamics of, you know, dispersal and sort of genetic distinctness in some of these populations. And so what I'm doing now is genome skimming. So we're pulling the UC loci that we need that will be compatible with that previous data set from just whole genome sequencing. And, you know, from that, able to improve greatly our sampling from some of these disparate localities that we didn't have a high emphasis on before. Next steps also include taxonomy. You know, again, going back, there are about 18 described species, and now we have even more than I thought before, which presents a challenge because one thing as we're finding in this group is that there's not a whole lot of diagnostic characters that can be found externally. However, internally, the insect genitalia can be very, very helpful for identifying these species, and so can these beetles' toes. So effectively, their claws. And you can imagine that if you're looking at a beetle that's the size of the L on a penny, its toes are much, much smaller, and it's quite a challenge to actually describe an image and do all this sort of work. And so anyway, I have to spend a lot of time looking at beetle toes and beetle penises. And you know what? My parents still talk to me, so we're all right. So yeah, you know, looking at this, why is not a micro so diverse? You know, it does look like the Guiana Shield is actually playing a strong influence in this, and that at least kind of gives us some directions. Now, moving forward, you know, I'm going to look at a group that kind of defines a little bit more my future, I think, here at LSU in terms of sort of this biogeographic and phylogeographic approach, and that is looking at the genus Supacellus. Now, Supacellus is a really interesting one, and because this group is probably younger than Notomicros is, and yet the morphological disparity and just the overall species diversity is like fourfold that of Notomicros. So this is all the same genus right here, and you can see just a lot of variation in the size, in the shape, color patterns, and I can tell you, even as someone who like looks at all the tiny little morphological bits, there's a lot of variation in all the tiny little morphological bits. What you end up with is about 70 described species that end up a lot of different widespread species groups. And so kind of for each of these little beetles that I have here on the right side, is there are anywhere from say 3 to like 10 species that they are representing, and a lot of times those species will actually get, or there will be members of those particular species groups that actually make it as far up north as Canada. So a lot of interesting questions to be asking each of these. I'm going to focus on one of these, this Supacellus nigrinus group, and kind of show you just how much this unfolds as you're looking in depth at it. So Supacellus nigrinus is very widespread in the tropics, getting all the way up into Cuba and southern Mexico and south to Argentina. But it's always been treated as just one species. The thing is that in terms of me doing some of my morphological approaches, I actually kind of noticed something. And so this is more beetle junk here, is that ED dissections showed really, really distinct differences in some of these individuals. In particular, I'll draw your attention here just to the cetal fringe on what would be the left lateral lobe, in which you have like a very sort of separated setose or hairy margin on one individual. You have a very kind of broad lateral lobe with a complete seedy row or row of hairs there. And so we saw these distinct differences and decided like, okay, we should probably maybe take a bit of a deeper look at that. And again, we're kind of approaching the same questions though. What is the extent of this overlooked biodiversity? And then, how is geography using this diversification in this group? And we decided to went ahead through some more UCEs at it. Because we used a Supacellus model as when we were building this tailored probe set, we actually got way more robust data capture. I mean, from a bait set that was targeting about 3,400 UCE loci, our 90% completeness matrix gave us almost 3,000. And from coming from an entomological standpoint, where we struggle for I spent a good part of my career struggling for 3 loci. I was pretty happy with this, and I figured I'd show it off on that. What we got from that was a very well-resolved phylogeny with congruent, highly supported topologies across all the data treatments that we did, and we recovered a total of 5 major clades in this group. Now, when we started looking at these different, some of these different clades and the individuals that we actually used in it, we found that, yeah, the external morphology is just as homogeneous as we thought it was. Like there's a reason that it was treated as one species, and it's because they all look like that. Like they all look exactly like that. But when we start pulling out these ediae and specifically the genitalia, you do actually see some morphology difference that are useful for delimitation and consistent with the clades that we found. The left lateral lobe again is indicative of clades, and in some cases even ancestry. And the median lobes, while wherever you find kind of more subtle differences in that left lateral lobe, the median lobes are kind of the main part of the beetle genitalia can still be informative. And so answer question 1: how many species are there? I'm not too sure yet. There's definitely more than one. And it looks like from this, there's about 5. Let's go for 5 for now. Let's not think too hard about it. But so meanwhile in Venezuela, though, I got scooped in notarids, like little tiny. A person, I do know him, works in Venezuela. And he went ahead and noticed this also and actually described several new species. I will say a little bit haphazardly, we do need to do some reconciling with some of these names, but it does look like he at least hit on some of the same things that we were seeing in our own research. And so it makes you wonder if we really needed to do all that data gathering at all for at least this question. But anyway, yeah. So we'll deal with that. But in the meantime, we can go ahead and talk about like some of the ways that geography is influencing this group. And if you go and kind of look at this geographic structure in terms of biology, like, for those of you that might be familiar a little bit with South American geography, you might see some patterns kind of come out right away. And that you see to have like, some groups that are kind of more coastal and kind of in the llanos of Venezuela, other ones kind of more in the uplifted region, and yet other groups that you'll find down in the basins of South America. So you do definitely see some geographic structure. But I wanted to look at this particular red clade in general, because if you were happen to notice, you'll see that these are all endemics very specifically, again, to this Guiana Shield region. And taking a closer look is all of these samples are from the savanna areas of the Guiana Shield. So going all the way from the llanos of Colombia and Venezuela, and across from the Ribbon, the savanna complex there, kind of on the borders of Venezuela and Guyana, the Sipilowini savanna that I posted a picture on, and finally going out to Amapá and getting into the more coastal. And just to kind of like remind you again, this is where I was here actually collecting some of these beetles that I used. And so we're starting to wonder is like, hey, do you actually see these savannas driving diversification or speciation? I mean, you have effectively sort of like an island biogeography question right here, right? These isolated savannas that during times of glacial maxima actually generally came together and were largely congruent. When the rainforests shrunk down into their refugia, these savannas expanded greatly and often became interconnected with each other. And so you kind of have this question is like maybe, by dispersal or by this sort of vicariant effect, you might see some genetic structuring. Again, we went ahead and pulled some SNPs. And quick stop right here. I'm just wanted to come and plug this. If you do happen to be messing around with SNPs and doing any kind of SNP filtering, my lab mate up at the Moyle lab at KU developed this really cool R program called SNP Filter. I have found it incredibly helpful for looking at SNPs and filtering SNPs from reduced representation data sets. It lets you kind of like interactively do this and actually look at like, what missing data cutoffs or minor allele counts and stuff like that do to your data so that you can kind of get the best SNP data set that you can out the other end. Anyway, plug over. So, looking at the Guiana Shield lineage, then, when we started looking at the phylogeny, it definitely does show some structuring across these savannas, especially with a particular east and west split. But then also within it is that you see the Sipilowini savanna kind of falling out as its own separate lineage sister to the Amapá savanna. And, kind of we end up with this like a little bit of a nesting here. But the thing is from the big, we were a little bit surprised to find out with how much savanna sampling we did in the llanos. Only one sample actually was found in this clade from the llanos of Venezuela. And we went ahead and we started doing some of these clustering methods. So the PCA reflects this split also with the east and west split most highly associated with the explained variants. Again, you can kind of see those all there. And then when we started running these phylogenetic networks, it was again just pretty consistent. You get an east and west split. You get that Amapá and Sipilowini split also there on the east end of it. And likewise, to some extent, with doing an admixture or a population assignment study, in which you get the east and west split as the preferred model. But instead of finding a break in the eastern populations, instead it was kind of showing up here in the western ones and in a very weird way. The take homes of this in general is that it's a very complicated story. Is that you're looking at, potentially not only patterns of dispersal and ancestry that are going to be reflected in these data, right, and in these SNPs that maybe it's either due to previous ancestry, it could be due to some type of recent gene flow or hybridization or just general ILS that kind of causes some of these patterns. And so it's a little bit difficult from this directly to tell exactly how these things are diverging, whether it's dispersal, and this is just like an isolation by distance thing, or if it's due to some sort of more complicated island biogeography type of effects as these savannas expanded and shrank. But in any case, we can definitely see that the savannas are influencing diversification in one way or another. And again, this is just sort of like a first step, and there's a lot more to do. So look in more detail at this whole story. So now kind of going on from there, I'm going to talk just a little bit about some of the emerging trends that we're actually seeing across some of these lineages. And looking especially at the geography, I was working with a student on some of this, and so I'm kind of asking, like, don't take photos or anything like that. What happened to be online, I guess. It's going to be on YouTube, isn't it?
It doesn't have to be.
No, we'll figure it out. I'll check in with my colleagues. But this is a project in which I was mentoring a master's student in my old advisor's lab, Andrew Short, and they put together this really good study where they found that, rather than looking at the ecoregions like we classically kind of think of it, we got that there is, some patterns that might reflect some of the ecoregions that you actually see, like, so for example, you see this definitely in, right, like the ecoregions in terms of if you're looking at sort of the dry scrub or savanna regions, you see some patterns of diversification being driven by an ecoregion effect. But a lot of it doesn't also work. And so they were kind of looking more at like, okay, what is, geology? Like how well does the patterns of the phylogenies and evolution that we're seeing fit the geology? And in this study that I wish I could show you the data, they're actually finding that there is a pretty solid fit with the geology rather than the ecoregions in a particular group, and neither the ones that I happen to be going over. And certainly, like, when you again looking at this, like that's not too surprising. If you look at the underlying geography and, kind of say if you consider Amazonia, right, that glosses over a lot of this underlying geological structure that might actually be influencing some of this diversification. And so it's not too surprising that geology might be the better predictor of at least a lot of deeper divergence. I was really excited when my student, not this one, that's not my student. You might know him. I was really excited when my student showed me these results, and we were putting the manuscript together. And at the time, me and Fernando, who was my longtime roommate and a really good friend of mine, were also sharing an office. And I turned around and I was like, "Hey, yeah, check out this pattern." And him and all of his cheekiness just kind of shrugged it, and he's like, "Yeah, dude, history's important." And then just went back to work. And I was like, "Oh, okay, well, well, I was excited." To some extent, we entomologists are still kind of like cavemen trying to catch up a little bit in some of this sort of stuff. And that, anyway, yeah. So I kind of always take that with me everywhere, and that really does kind of a lot. Is that, yeah, geology is probably better captures the actual history of a lot of these groups. So to kind of recap, the drivers, vary widely across a lot of these, clades in different scales. Some similarities you might find apparent between these different groups, such as Traili versus the Negrinus group. But then there's also a lot of differences, and so there's still a lot to kind of parse and understand from these different clades. And so kind of, yeah, I realized I was trying to put together, okay, like what's the actual take home of this talk? And I realized, hey, it's complicated. There's different patterns emerging at different scales of evolution. But I realized, well, yeah, we know. Like, it's not really that profound, but it somehow feels profound. I don't know. So I kind of want to close the talk a little bit and just talk, very briefly about how this relates to the work that I'm doing here at LSU and with the Louisiana State Arthropod Museum. And I thought the best way to kind of bridge the talk was just to show you by the same person, Sean Conway, just does these wonderful 3D renders of all these maps. And this is looking here at the United States. And, I'll kind of draw your attention here to Louisiana, or at least this general Gulf region. You almost kind of see some of the same stuff. You have this basin surrounded by uplifted regions. And so it kind of gets, your brain working a little bit on how some of what I've been doing might actually be, pretty applicable. And so sort of what I'm approaching with this idea is not only looking, at South America, sort of look more in North America, and not only at notarids, but at a lot of different groups too. And just in general, sort of build my lab around this idea of using aquatic beetles as sort of a model for comparative investigations of biodiversity and evolution. Those are both just a couple of quick examples of that. Now my students, and again, like I'm looking here at this Suphacelis phylogeny. And just to kind of remind you that, I showed you this pattern that was unfolding just by looking at this tiny little piece, and to remind you that no one's really looking at any of these at all, except us and my grad student, Jacob, who is now kind of, devoting a couple chapters of his dissertation on building a robust phylogenomic phylogeny of this group, and kind of to start looking at, patterns that might exist in parallel across all these things. You have this really nice monophyletic group exists only in the New World, and then has all these nice sort of independent diversification events or patterns kind of happening up in its tips. And so I think it might be a really, really cool place to study. Likewise, I have a Lagniappe scholar that's working us right now and putting together the first phylogeny of this group called Liocanthydrus. This was the very first paper I ever published, a genus revision of a stream endemic from South America. And then finally, I'm trying to find some ways to sort of just keep things a little bit local. And so this is a really cool project I'm getting more and more excited about as the findings come up. Is that, you know, this giant mess that is going on right now with the University Lakes project, right? They're dredging it, they're rebuilding all the shores. And so we, through some input from one of the other professors in the entomology department, thought it would be a good idea to go ahead and start setting up some baseline data on the aquatic insect communities that are there. And so this summer we went ahead before they destroyed all the shores and went and started just doing some collections to kind of see what the aquatic beetle diversity looks like, though we are collecting all arthropods, or at least as many as we can. And then that way setting up kind of a baseline that as, you know, this dredging stops, the turbidity drops, and, you know, the shorelines actually recover, to sort of track any sort of changes that might be happening in the community over the next several years. And it's just kind of a nice project that, you know, as undergrads come in, can kind of pick up, start adding more data, make different contributions, and you know, maybe just start really amassing a cool little data set right in our own backyard. Why not, right? I think it's pretty cool. And this is being done by, headed or led by Riley Norton, undergraduate in our lab. Managed to even recruit some folks to go and help her. So that was nice. Likewise, I'm finding that my both my field component of my research and then the genetic component of my research is applicable in a lot of other places. And so I'm starting to get into forensic entomology randomly. I never saw that one coming. Where I am going, and we basically made a body farm up at one of the local research stations. In this case, got the Louisiana Department of Wildlife to donate a bear to us, and went and just kind of like throwing all these animals out there, collecting beetles off of them so we can study the metabolomes of particular groups. And then also doing some, at least trying to kind of get, you know, getting into the sort of the ag center focus of serving Louisiana. In this case, sort of using my genomics background to start developing some eDNA approaches that might help us detect, you know, invasive species or crop pests, or oftentimes it's both invasive crop pests like this one right here that just popped up. So we can do, like, you know, really effective pest mitigation, or at least do more effective monitoring. I'm really excited because this is also, I think, very translatable to just community monitoring in general. And so hopefully, you know, got a seed grant for that. So really hoping that gets some legs, or it's getting legs. I am sewing legs onto it. And so, yeah. And, you know, I can't thank the folks at LSUMNS and Dr. Chris Carlton, really, really helped all this, really support. Naturally, as I mentioned, you know, I come from a museum background. It's really awesome to be the director, or, you know, of this whole museum, and really kind of guiding not only the way that it's into my research, but just in general and how it like serves the department and stuff. And so that's been a lot of fun so far, and you know, really value all the support from both Victoria and Chris and the rest of the LSAM team that's been there to help us out. And I apologize, a little bit of an older photo. Our team has grown a little bit, and not everyone's represented there. So you're in the audience and you're not there, I apologize, but I love you just the same, I promise. So yeah, I'm going to go to kind of leave it there, kind of with this last, you know, image, and thinking about these independent invasions of water, you know, where you might, and kind of like where I'm going to be looking at now in our own backyard as well as abroad. You know, be happy to thank everyone that helped me out with this. I you know, really have to thank, I didn't mention this at all, but I was like an ornithologist for like 3 years. And I had a whole project and everything. But, you know, they were all very, very helpful. Andrew Short, my advisor, has just, you know, bar none, but a huge influence on me, and really has given me the foundation, both in education, but also even helping me out with all the field work and specimens to do all this. Rob Moyle in the Moyle Lab, you know, Devin Durrat and Lucas DeSico especially, you know, were really helpful in allowing me to parasitize their lab for methods that I might apply to my own data sets. And I appreciate that also. So yeah, thank you, everyone. I'd be happy to take any questions that you might have. Yes.
You mentioned things to look at when you're trying to steer out species totally makes sense. I get it. But what is that? Do you think they're under central selection as well, or is somehow scale around that's causing a lot of diversity?
Uh, I kind of wonder about that too. So the question is you know, that I'm looking at beetle peepees and their toes, and that, you know, their genitalia, you know, being involved with reproductive isolation and therefore kind of making sense, right? That you would find differences there. And the question is why are we looking at their toes? And I don't know, for some reason there is sexual dimorphism, so you only find males that have the really distinct differences in their toes. I have You see this a lot across aquatic beetles, in particular in this group Adelphiga. So in the greater suborder, it kind of comes and goes. Uh, Part of me thinks a little bit it might have to do with grasping. You definitely see a lot of modifications in a lot of aquatic beetles for grasping mates during copulation. But another part, I kind of wonder if it doesn't have to do with just development. Because you like, if you think of like the genitalia are ancestrally appendicular, that it could be that by changing the genitalia, you end up kind of changing some of the development of some of the tarsi too. The only reason I think that doesn't make sense though is because you only see it in like the fore and mesotarsi. You don't see it in the hind legs or anything like that. That could be some other interaction, but who knows?
You know, they're being used in portion, which is what's making the genitalia go through places, you know, trails.
Yeah, no. So female role, and you see that in Dytiscids elsewhere or other aquatic beetles within this group. Is like, so I didn't show this, but my beetles actually have suction cups on their protarsi, and they use those to hold on to the females during copulation. Now, a lot of these aquatic beetles end up practicing mate guarding, and because they breathe atmospheric oxygen, what happens is by having a mate on your back for up to like 24 hours, it comes at quite the fitness cost, right? And so in a lot of these groups, you actually see the females developing cuticular modifications on their pronotum and on their elytra, so that kind of dorsal shell of their body that prevents the suction cups from grabbing on. And it's actually causing, you see it in some groups, actually causing like a coevolutionary arms race where you see females with really, really intricate structuring on their dorsal surface. The males of that same species will have very, very complex like array of suction cups. They have a bunch of big ones, they got a bunch of little ones, a bunch of medium ones, and really, really huge pads in which they can. And so it's not, you know, it's not too far beyond the pale to think that that might be happening. I wonder a little bit, just because of how small their toes are, is like I don't know exactly where they're sticking those toes. For probably, it's a weird. Anyway, I don't. Yeah, I don't know where those toes are going, so I can't really speak to how much they might actually be used in mate grasping. Yeah, something.
Nice thought.
Thank you.
I also find it quite amazing that you got students working on beetles as one that happen.
Yeah.
That first group you talked about,
Notomicros, yes.
You did get them to 13. So how do you get this long distance dispersal between all the world and what's?
Oh, I apologize. So that was the date of the specific group within the Neotropics I was looking at. Yeah. The whole genus, I think we dated to about 90 million years. If I remember right, in the divergence time, we actually had that date. So the old world and new world crown age is like 90.
But you still need a long first because that's still post.
So the crown age predates Gondwanan vicariants. So that I might have a. Oh, there we go. I can actually tell you. I have a slide up here. So the crown age of the new and old world groups together came out to just about 75 million years, but the lineage divergence from its most closest related sister, Phragodites, goes all the way back to looks like about 110 million years. And so this group, at least at the global scale, is quite old. And then the New World clade is a little older than 60 million years. And then within that particular trail group I was looking at was 13. And so the long distance dispersal was basically from, maybe not necessarily dispersal, might have just been vicariance of happening through like South America, Antarctica, and then Australia into the Pacific that way. Does that?
Yeah, I'm just wondering if there's some mode of dispersal for crossing even oceanic, like are they hopping on birds? Are they?
I see.
Even in the neotropics, they're still moving quite distances that are, for something so small.
No, yeah, good question. I apologize. Yeah, so how are they dispersing at all? Well, they can fly for one, which helps out quite a bit. But I will say, kind of, related to your question, is that like you find these things in like Fiji with relatively recent divergence from some of its like some of these species actually have up here this punctulatus species up there, kind of towards the top. Is a Fijian species. You find it out there kind of way like all on its own. And so, yeah, I got there somehow. How it got there, like I had no idea. They're so small. If they can get airborne, maybe they're just kind of blown around. I really don't know for sure to answer your question. At least they can fly though. So that at least gets them up and out of the water. Yes.
Really cool and interesting system. I have okay. What kind of DNA use were you getting from those 2 millimeter beetles?
Yeah. So the question is how do I get DNA out of these things? I will tell you that, especially when it comes to the genomic side of things, you basically get one shot at a library. So far, out of Notomicris, I've been able to max out at about 100 nanograms, and that is when I smash the specimen, go ahead and throw it in a mixer for like 24 hours, and then try and take as much DNA as I can.
From one individual?
From one individual. And yeah, we're able to do that. It's not much, but like, the library preps are doing now, for example, we can do with 50 nanograms or even less if we need to. And their genomes are only about 200 megabases. And it actually makes this so efficient to just go doing the genome skimming thing. Because I can throw these on like an Illumina lane. And yeah, I mean, you can pile like literally on the NovaSeq, like 25B lanes, like hundreds of them if you want to, and actually get good data from. So yeah, that's helpful. Yeah, but it's a challenge. Some of the beetles I did, there was one, let's see, Maruidi, which is like the size of, I don't know, like medium grit sand or something like that, like, 0.8 millimeters roughly. I haven't been able to get more than 20 nanograms total out of one specimen. And I sent libraries out using only 10, and it was like one of our best assembled genomes. So it was 110 megabases. So I'm kind of actually curious about that, is that as I'm pulling these genomes, there seems to be a size correlate with genome size, which is not necessarily something you see elsewhere all the time. And so I'm a little bit kind of curious about that in this group. And it's kind of another, we started working on it a little bit in the Moyle lab. And it's something I hope that we can eventually kind of keep on pursuing, is looking at the actual just genome evolution in this group. There's like, things like, what do you call the sex determinism, for example, changes a lot in this group. And so there's a lot of questions to look at genome evolution just in itself.
Have you tried to take DNA from specimens in collections, like ancient DNA or something like that?
I haven't done any, short answer, no. I've done museum specimens, but that's about as far as I've gotten there. I'm trying to stay away from ancient DNA as much as possible. eDNA, I'm all right with it, but I had a lab mate or an office mate that did that at KU, and I did not envy her. Cool research though. Yeah. Yep.
That was great seeing it. So they're morphologically conserved. Are they doing pretty much the same thing ecologically? Are there differences in diet or microhabitats or anything like that across those areas? They all look the same.
A good question. Are they, being so morphologically conserved? Like, are they also ecologically conserved? Right. And short answer is yes and no, is that you have, most of them are mostly doing the same thing. But you do have a bunch of like, for example, in Notomicros itself, in one of the other lineages, we actually have a transition back to the terrestrial habitat. So we have a species that we only find in leaf litter that we think is parthenogenic, because I cannot find a male for the life of me. But you only find them in leaf litter. And that seems to be happening elsewhere in both Suphacelis and Notomicros, where I think there are some shifts to parthenogenesis, because there are some where you get just vast series, hundreds and hundreds and hundreds of females from all across their distributions, can't find a male to save my life, or you find like one. And so it might be like a facultative kind of thing going on, or, rather than like an obligate. Then there's some other ones like, within that Notomicros trail group, you actually had was where Petroreptans was at. So that was the rock seep specific one, while the rest of them you're finding kind of in more forest pools and kind of the margins of very small creeks and stuff like that in leaf packs. And so there's definitely some transitions happening in the group. There's one species also in the Phragodites, the one at the very top of this, that's sister of the rest of the group. That is a subterranean beetle. And there's a couple of those in this group also. So you find those in aquifers in Japan. Yep. Yep. Yes.
Logistical question. So you mentioned some cool results from Venezuela.
Mhm.
You also said you're not destructively sampling collections. Does that mean you got to go to Venezuela?
I did not go to Venezuela. Unfortunately we stopped doing that right around when I went to grad school. I went all the other areas around that. So we did, Guyana, Suriname, French Guiana, Peru, northern Brazil, all that sort of stuff. Either me or my advisor. But my advisor was the one that was doing it. Sometimes I just went with him. And now I'm kind of doing Peru stuff. But so the short answer is no, I didn't get to go. But there was a lot of field work happening there before 2012. Yeah, it's a bit of a hard place to get into.
Any last questions?
Thank you all
Let's
again.
give one last round of applause.